Hello, and welcome to part 2 of chapter 4, covering the very broad subject of molecular biology. Today I'm going to talk to you about DNA replication, mitosis, or the cell cycle, and just a little bit about inheritance. So I'm really just going to scratch the surface on these topics because we really could go into detail. But that really could just be an entire class in itself. So I really want to try to keep this as simplified as possible. All right, so let's start off with this. Let's begin with DNA replication. So before a cell divides, we have to duplicate our DNA, all right, so that we can have a copy of our genome sent to the daughter cells of that original cell. So what are we talking? We're talking about how one cell is going to divide into two cells, or daughter cells. And then those daughter cells will also divide into two cells, and so on and so on and so on. So in order for the new cells to be complete, we have to have our genome sent to the new cells, right? So before we can do that, before we can actually have a cell divide into two cells, we have to make a copy of the DNA. In other words, we have to duplicate the DNA. This is also called DNA replication. And, you know, all of this follows something we've already talked about, known as the law of complementary base pairing. So basically, we can predict what a sequence of bases is going to be on one DNA strand if we have the other DNA strand. Remember that the DNA molecule is a double helix. It is composed of two strands which are complementary to each other. And so, you know, this, like we've talked about before, kind of like how we talked about with RNA, how RNA makes a copy of DNA. Well, this DNA replication basically follows the same pattern, where we're going to add bases to a growing strand of DNA to make another strand of DNA. And we'll see how that happens. We'll see this in pictures, and I think it'll make more sense that way. So DNA replication really can be divided into various steps depending on how deeply you are studying it. For our purposes, we are just going to break this down into 4 categories and just stick to the main idea of what's happening in these main categories. All right, so we're going to break this down into 4 steps for DNA replication, which you definitely want to know about the 4 steps, at least just as far as we're talking about them. So first of all, we're going to have our DNA unwind from histones. Remember that when we have our DNA stored in the nucleus, because the DNA molecule itself is so long, we have to sort of condense it in order to have it fit into the nucleus. So the DNA will unwind, or I'm sorry, it will be wound around histones, right? Remember that DNA will sort of adhere to the histones, and this will end up causing the DNA molecule to condense. Now, when we're doing DNA replication, however, we're going to need to get access to that DNA because we need to copy it. So the first step in doing that is that we have to release that DNA from histones or end up uncondensing it. So how do we do that? Well, we're going to break down the histones. If we break down the histones, then the DNA has nothing to adhere to. All right, so this is a temporary event that's really only going to happen during DNA replication, because normally we want our DNA to be bound to these histones. So when we're trying to copy the DNA, well, we're going to unwind DNA from histones by a process that you already know about called hydrolysis. So as long as water is present, water will come along and interfere with the chemical bonds existing in histones, and then this will cause a breaking apart of the histones. And now after I talk about these steps, we're going to see a picture of all this. Okay, so once we have unwound the DNA from histones, then another event is going to take place. We're going to see this very important enzyme that y'all need to know. It's called DNA helicase. DNA helicase is going to come along and unzip the double helix. This is why we call it unzipping. Okay, we're basically separating the two strands that make up that DNA molecule. And this way we have exposed the nitrogenous bases existing in both of those strands. Okay, so DNA helicase is going to serve to unwrap or unzip the double helix. Where this is occurring, we call this a replication fork. Okay, this is basically the point at which we have two single strands instead of the double strand of DNA. The next step is that we're going to have another important enzyme. This is called DNA polymerase. DNA polymerase, you want to compare that to another enzyme we've already talked about called RNA polymerase. Remember, when we talked about transcription back in part 1 of this chapter, that I mentioned how RNA polymerase is basically going to build an mRNA strand based on a DNA template, right? So RNA polymerase copies the DNA, forming a complementary strand of the genetic code, and we call that mRNA, messenger RNA, which will eventually leave the nucleus and go out and do other things. So DNA polymerase is very similar to RNA polymerase. All right, but in this case, DNA polymerase is building a new DNA strand, right? RNA polymerase was building an mRNA strand. DNA polymerase will build a new DNA strand. Okay, so these two enzymes work very similarly, but when we're talking about DNA replication, it's DNA polymerase. So yeah, just like I said, like RNA polymerase does, DNA polymerase will read the exposed bases on DNA and add free nucleotides. Remember, there are just free nucleotides floating around, and so DNA polymerase will add free nucleotides to a base that is existing on the strand it is basically copying. Now, what we're going to end up seeing is that there will be two DNA polymerase enzymes working on each strand, all right, of the DNA. Remember, DNA, it unwound from the histones. DNA helicase came along and unzipped the double helix, so now we have two strands separate from each other. And so each of those separate strands will be worked on by a separate DNA polymerase. And you end up having copying of each of the DNA strands in opposite directions. And again, we're going to see a picture of this here in a minute. Eventually, what you end up with is you get two daughter DNA molecules that were made from the original parental DNA. So the two daughter DNA molecules are complementary to the parental DNA code. We call this semi-conservative replication. All this simply means is that each daughter DNA is consisting of one old strand of the DNA, one original strand, and one new strand. And so those two strands will then come together to form a double helix for that new daughter cell. Finally, we will have the newly made DNA repackaged. Now, this first point here is just really for your interest. You will not be asked about this. Again, I just left it in case you're interested. You know, at any given time that we're doing DNA replication, there will be thousands of DNA polymerase enzymes working at the same time on DNA. So this is really happening at a very high rate, very rapidly. And again, you know, you don't really just have two DNA polymerase enzymes. You have a lot of them, thousands. And this is, you know, for the purpose of speeding things up. We don't want this to take forever, right? We don't want cell division and mitosis to take forever. So we're going to do this pretty quickly. And yeah, as it says here, all 46 chromosomes, we have 46, right? All 46 are replicated in only 6 to 8 hours. Okay, and again, I'm not going to ask you about those details, it's just there for your interest. So we have our newly made DNA. We have our daughter DNA. Then what? Well, we need to repackage that DNA. And so, of course, we're going to need to wind this new DNA around histones once again. And, remember, we broke down those histones in the first step. Well, all this while that DNA is being replicated, all during that time, we are in the process simultaneously of making new histone proteins, right? Histones are proteins, so they're made in the cytoplasm. Once those histones are made, they will be transported back into the nucleus, all right? And so then the histones will be there and ready for the DNA to wrap around and become condensed again, and there we have it. Okay, so we'll just do the same thing. We'll wrap that the DNA helix around the histones to make up the nucleosome. And then here is your picture of all this. You can see everything that I talked about. So in the very beginning here, where on the left where it says parental DNA, now what's happening here, and it's actually not showing it in the picture, but you're having the unwinding process happen here. Remember, the unwinding process is relating to how histones are going to be broken down by way of hydrolysis. All right, and so again, that is not specifically shown here, but you should know that that's going on. Once we have our histones broken down, that's when our very special enzyme called DNA helicase is going to come in. And DNA helicase is responsible for unzipping the DNA molecule. So you can see here that the double helix will be unzipped, okay, or it will be opened up so that now you have two individual strands. DNA helicase is responsible for that unzipping or that separation of the two strands. So you'll end up with one original DNA strand up here, and you also have another original DNA strand down here. Notice that this area where we have this unzipping start, this is called the replication fork, and that is a term that you should know. The replication fork, it's kind of like when you see a fork in the road, right? You're walking on the street, you're walking on one road, and all of a sudden you get to a point where the road veers off into two or more separate directions. All right, you call that a fork in the road. Well, that's kind of like what this is, right? It's a replication fork. One original strand up here, the second original strand down here, and DNA helicase is the one responsible for that. Then what? Well, then we're going to have our other really important enzyme involved in DNA replication come into play. That is called DNA polymerase. So DNA polymerase is represented by these big green structures here. All right, these are enzymes, DNA polymerase. Remember, anything that ends in ASE is an enzyme. All enzymes have that ending. So yeah, DNA polymerase, what is it going to do? Remember, it's very much like RNA polymerase. Okay, but in this case, we're making a new DNA strand, so we call it DNA polymerase. It's going to do just like what RNA polymerase did. And it is going to add nucleotides that correspond to whatever nucleotide or whatever base is on this original strand. Okay, so if, let's say, on this original strand, this DNA polymerase is reading the nucleotide thymine, well, then it will grab an adenine, because remember, thymine and adenine go together. All right, it will grab an adenine from the pool of nucleotides and add that adenine to the thymine. This causes a bond, right? This is a bond that's taking place between the two bases now. And then we end up with a new DNA strand. We end up, again, with a double-stranded molecule, where one strand was the original parent DNA, and there is a complementary new strand that is being formed based on whatever bases the DNA polymerase reads on that old or that original DNA strand. Okay, so your daughter DNA is going to consist of one old strand that came from the parent and one new strand that is a complementary version of that old strand. All right, so, and again, to speed this process along, we're going to have many DNA polymerase enzymes, all right, working on our DNA. So this DNA polymerase at the top is moving in this direction. All right, it's moving in that direction as indicated by the arrow. What about your other original parent's DNA? Well, same thing is going to be occurring on the other strand, all right? Again, this maximizes the speed at which we can do this. So we're simply going to have another DNA polymerase molecule, or enzyme, working on the second strand of DNA. And the same thing is going to occur down here, all right? And that really is the gist of it, okay? And so once we get our new strand of DNA, we're going to need to repackage it. That's the last step. And, you know, the DNA will then wrap around the new histones that were made and all that. Now, here's an enzyme down here I didn't mention called DNA ligase. And we're not really going to deal with it. I mean, basically, a ligase is going to be an enzyme that sort of like ends something or cuts. To ligate means to cut. So DNA ligase is actually just going to sort of sever the strand there. I'm not going to ask you about it, though, because we didn't really talk about it. All right, so really, this is the gist of DNA replication. All right, as long as you just know it to that extent, you're going to be just fine. So now, of course, mistakes can occur along the way. Errors can occur, all right? But there are going to be many checkpoints along the way. And towards the end of this video, I'll mention a couple of time points at which during the cell cycle we're going to have what you call checkpoints, okay? But there will be multiple errors, okay? But again, we have these checkpoints, and there are actually multiple enzymes that can come along and correct any kind of errors that might occur, okay? DNA polymerase is one of those enzymes. So not only can DNA polymerase add new nucleotides based on a original parent DNA strand, but DNA polymerase can also check to see, are the new base pairs correct? If there is something that is incorrect, maybe a certain base was not supposed to pair with another base, well, DNA polymerase has the ability to correct that by taking off whatever was incorrect and adding the correct base. All right, so DNA polymerase can do this, can fix its own mistakes. And it's so good at it that actually, you know, as the statement says down here, that you may have typically only one error per 1 billion bases replicated. That's really the end result, because DNA polymerase and some other enzymes will work to prevent these errors, okay? But keep in mind, you may still get an error, all right? And it could be an error that causes a disease, all right? And actually that is the case for many different types of diseases that have a genetic basis. Now, let's mention mutations. What is a mutation? A mutation, very simple definition for this. It is simply a change in the DNA structure. That's all we can say a mutation is. It is a change in DNA structure. What causes that change in DNA structure can be caused by many factors. All right, it could be environmental factors like radiation. Viruses can cause mutations. Certain chemicals can cause mutations. Or maybe DNA polymerase or the other enzymes that at our checkpoints didn't fix the error. All right, so there are several ways that you can get a mutation. Now, what's important for you to know about mutations is that some mutations may not cause any effect in the organism at all, virtually no effect. Other mutations might be detrimental to an organism. Maybe this mutation is going to end up being damaging to cells and tissues. And guess what? Cancer. I'll just tell you this. every single cancer that exists, every single one, is caused by a mutation. The question is, what causes that mutation? All right. Another cause to mutations is, it's kind of like, you know, when you age, you're kind of like an old car. All right, you start to break down, and the engine doesn't work as good as it used to. All right, and what ends up happening is that in older age, you're not as good at DNA replication as you used to be. As you age, you get more and more errors here during this, somewhere in this DNA replication process. And so this is why you tend to see cancers happening in older people, all right? Because older people are more prone to having these DNA mutations. And like I said, all cancers, every single one of them, is caused by a DNA mutation. Again, the question is, what's causing the mutation? It could be in old age, where just this replication process doesn't work as well. Or it could be due to exposure to something. All right, radiation, viruses, chemicals. All right, there are many different chemicals that can lead to cancer. All right, example, asbestos. Everyone's heard of asbestos? This is actually a type of lung cancer that affects the mesothelium, which is a lining of the lungs. And how was this caused? It was caused by inhaling asbestos, which is something that is found in really old buildings. So you found that this big surge a while back of a bunch of people working in the construction business getting this mesothelioma cancer caused by the inhalation of this dangerous asbestos. All right, but there are many examples of this kind of thing. Also keep in mind that mutations, all right, I said mutations may have no effect, they might have a detrimental effect, or you can have mutations that are beneficial, all right? You can have what you call beneficial mutations. So a beneficial mutation would be something that is going to allow an organism to survive better in their environment. I mean, there are a lot of examples of that too. And I'm just going to give a quick one. Think of zebras. Zebras out in the wild. Do you know that the zebra stripes are actually mutations? They are genetic. That is a genetic mutation. That is a physical trait that resulted from a genetic mutation. But what ended up happening was that this mutation was beneficial to the organism. It allowed the organism to survive better, because with stripes, the zebra, if they're in big groups, if you've got hundreds of zebras all together, well, if a lion is coming along and trying to hunt one of those zebras, that lion has a real hard time picking out the zebra, because all the stripes on all those zebras make it really confusing. The lion cannot pick out a single individual zebra because it all just meshes together. So that's kind of, an example, at least in non-human animals out in the wild, of a beneficial mutation. So keep that in mind. All right, mutations can be advantageous. They can be dangerous or detrimental, or they may have no effect at all. Okay, so with that, let's move on to the next part, the next topic of this. We're dealing with the cell cycle. So this is very much a summary of what is presented in lab. All right, so lab 4, the second half of that was dealing with the cell cycle and mitosis. So this is all just sort of a repeat or a summary of that. So the cell cycle, we're talking about how the cell is going to divide, basically. There are two major phases to this. You have interphase and mitosis, or the mitotic phase. And you can break these two down into subphases. So interphase you can break down into G1, S, and G2. Then we go into mitosis, which can be broken down into prophase, metaphase, anaphase, and telophase. We also can break two of these down even further. We can break prophase down into two phases, early and late prophase. And the same thing with anaphase. We have an early and a late anaphase. And actually there's another small stage here which I didn't include, and it's called prometaphase, which occurs before metaphase, but I decided just not to include that. Here's a schematic of the cell cycle of all these stages. You will want to know a little bit about what's going on in each stage, and we're going to go through it. So first we have interphase, which is represented by this green arrow. All right, so most of the time your cells are in interphase. Interphase we're going to break down into G1, S, and G2. G1, I usually think of G as standing for growth. All right, so you will have just a series of development and growth taking place in G1. And growth, I mean like development of organelles and just normal metabolism going on in the cell. After G1, you will have the synthesis phase, and this is where we're having DNA replication, which was just described to you. Then the last stage of interphase is called G2. And again, this is just more growth occurring, and you're going to get an increase in metabolism during this phase because we're starting to prepare for mitosis at this point. After G2, you will then enter mitosis. And so then we will encounter prophase, metaphase, anaphase, telophase, and then cytokinesis, which I think I forgot to mention on the last slide. Okay, but we're going to go through all these. So interphase, I already mentioned these, right? G1, S, and G2. G1 is the first growth phase. And this is where we're having growth and accumulation of items in the cell. And then S phase is where we're having DNA synthesis or DNA replication. All right, when you say DNA synthesis, it's really, you're also saying DNA replication. It's basically interchangeable terms. All right, yeah, so during S phase, we're replicating all of the DNA that's in the nucleus. G2 is the second growth phase, okay, and we're continuing to have growth. We're checking for DNA replication errors. We are growing and synthesizing enzymes that are associated with the next phase. All right, so G2, we're continuing to grow. We're checking for DNA replication errors, and we're getting ready for mitosis. Okay, so then after interphase, we'll have our mitotic phase, sometimes called the M phase. All right, so we're going to go through the stages of mitosis. All right, and remember again, mitosis is when we are dividing one cell into two genetically identical daughter cells. And again, a reminder of when are we going to be doing mitosis, or, you know, what is the purpose of mitosis? Well, obviously during development, all right, after, you know, from conception up until, you know, the end of adolescence, your tissues, your cells, your organs are in the process of developing. All right, so you need mitosis to be occurring. Also, replacement of cells that die. Now, sometimes we have like what you call normal cell turnover. We will talk about this when we go into the skin. When we talk about the integumentary system in chapter 6, we will mention this, all right? Basically, like in the skin, you have cells that die. This is a normal process we're continually losing or shedding our skin cells, and that's normal. And so we will have mitosis in the skin occurring all the time to replace those cells that get sloughed off or that are shed. Another reason we're going to be doing mitosis is during wound repair. So if you have an injury, right, if you get some damaged tissue, we're going to want to replace that tissue with new tissue. So this is mitosis. Now, not all tissues can be repaired. Later on, we'll talk about that, about like what tissues do regenerate after injury. And yeah, there are some tissues that do not regenerate after injury, and we'll compare and contrast those at a later date. All right, so our phases of mitosis, we've got to know these. Now, keep in mind, for each phase, you're going to want to know a little bit about what's going on in each stage and be able to recognize what each stage looks like from a model. Now, that's really going to be for lab. So in lab, you will look at these models and try to identify what stage is being shown. So let's go through each one here. Prophase. Now, I think I may have been unclear previously about this aspect, but basically in prophase, we are going to make sure that our chromatin is in the form of chromosomes, just because they're easier to distribute to the daughter cells later on. Okay, so during prophase, we will have our 46 chromosomes. All right, remember, DNA has already replicated. DNA has already replicated. And so we have 46 chromosomes, and each chromosome is made of 2 chromatids. Now, what's happening in prophase is that we're going to see the nuclear envelope disintegrate. In other words, the nucleus is going to start to break down. Why do we want that? We want that because we need to get access to the genetic material that's being stored in that nucleus, because we need to eventually pass that genetic material on to our daughter cells. We will also see during prophase that centrioles, remember centrioles, these are organelles, okay, and here is where a big part of their function comes into play. Centrioles are going to start sprouting microtubules. You remember microtubules we talked about in chapter 3. Microtubules play a very important role in mitosis. Now, sometimes you might see these microtubules referred to as spindle fibers. I'll probably not call them that. I will call them microtubules or the mitotic spindle, because what happens is these centrioles sprout microtubules, and then the microtubules will end up forming a more complex structure called the mitotic spindle. So what's going to happen is the microtubules, as they grow, remember that microtubules can assemble to become longer. And so that's what's going to happen here. Microtubules will grow longer, and as they grow longer, they're going to end up pushing the centrioles apart, so that the centrioles will end up on opposite sides of the cell. All right, we can call these opposite poles of the cell. All right, so microtubules are pushing centrioles further and further apart from each other, so that eventually the centrioles will end up on opposite poles of the cell. Now, there are other microtubules that play a different role. Some microtubules are later on going to attach to the kinetochores of centromeres of chromosomes. Do you remember these terms from part 1? Okay, as a reminder, we have chromosomes. Chromosomes are made of chromatids. Okay, so you have 2 chromatids per chromosome. Now these 2 chromatids are held together by something called a centromere. Now on the outside of the centromeres, you have these other little structures called kinetochores. It is the kinetochores that the microtubules will attach to in order to achieve the process of moving chromosomes to opposite sides of the cell. Okay, so microtubules have a dual role here. Microtubules will push the centriole pairs apart, pushing them to opposite sides of the cell. And then later, you will see that there will be other microtubules that will attach to the chromosomes at their kinetochores in order to basically move these chromosomes around. Now remember, prophase has 2 stages. We can break prophase down into early prophase and late prophase. So what's different about them? So in early prophase, our centrioles, okay, what we're seeing during early prophase is we're seeing basically the development of a mitotic spindle. You're seeing this start to form. And that's really what characterizes early prophase, is just the development of this mitotic spindle. And so a couple of other terms here I want to clarify. So centrioles, you already know what those are. Those are actually organelles, right? And it's from the centrioles that we will have our microtubule sprouts. And then once we have this occur, you can actually refer to the centrioles as something called centrosomes. I don't want this to get too confusing for you. Okay, but basically centrioles will further develop into a sort of a modified structure known as a centrosome. Okay, so all a centrosome is it's a modified version, or it's a it's a modified version of a centriole. Okay. And so the centrosomes will end up being at opposite poles of the cell by the time you end all of prophase. Okay, so what's the idea here? Mitotic spindle is forming. Okay, that's what's happening in early prophase. And again, we're going to have other microtubules with other functions here. So there will be these really different microtubules that are actually radiating from the centrosome, and these are called asters. And I'll point these out. Okay, so development of the mitotic spindle with a couple of details here for early prophase. Late prophase, what really marks this stage is that this is where you really see the end of the nucleus. All right, you see the nucleus breaking down. All right, and at this point, we're going to start to see microtubules attaching to the kinetochores of the chromosomes, which are at some point going to start moving these chromosomes around. Okay, but first we need to get our mitotic spindle fully formed, we need to get our nucleus broken down, and we need to have these microtubules continue to push against each other as they grow. Remember, as the microtubules grow longer, they're pushing on each other, and they end up pushing the centrioles apart. Okay, so what really characterizes late prophase is that you're now starting to see these poles moving to opposite sides of the cell, and we have our nucleus breaking down. Here's a visual of early prophase. On the right here, which has a lot more detail, on the right, okay, early prophase, we still have our nucleus. All right, we have our chromosomes consisting each of 2 sister chromatids. Remember, remember your terms, right? Your definitions with all this. Centromeres are going to be the little structure that's going to hold the sister chromatids together. So we still have this structure here. We still have a nucleus. So what really marks early prophase is the development of the mitotic spindle. Now up here we're seeing these like rectangular type structures. These are your centrioles. And from the centrioles you have microtubules that are sprouting out from it of each. Okay, you're going to have basically two sets of centrioles. One here, one here. And from each you will see microtubules extending out from them. These microtubules are going to be growing in length. And as they grow in length, they push against each other right here. And that causes the centrioles to push apart. Okay, so that we're eventually going to see them form centrosomes here and then up here. Now on the other end of these centrioles, you have more microtubules. These look like little shorter microtubules. And they form like sort of a star pattern around the centrosome, or around the centrioles. And this is called an aster. Okay, so basically when we have our centrioles and we have these little microtubules stretching out from this other side, then we can call that an aster. All right. An aster is basically a centrosome with these radiating microtubules. Here is like an actual cell, right? We have a nucleus in blue. And yeah, so we still see the nucleus in early prophase. But what you're seeing here to the left of this cell is how this mitotic spindle is forming. So how is early prophase different from late prophase? The most remarkable change in late prophase is going to be, well, there's no nucleus now. The nucleus is disintegrated, and now our chromosomes are free. And so now what we're going to see is that the microtubules that make up the mitotic spindle are now going to attach to the kinetochores that we're holding, that are located on the centrosomes, right? So this is how the microtubules are going to attach to the chromosomes, is by way of the kinetochore. All right, and then so at this point we're going to see this connection. We're going to see that the microtubules are attaching to the chromosomes at the kinetochores. And so now what we're going to have happen is we're going to see that these chromosomes will start moving based on microtubule movement. Metaphase is the next phase. This one's really easy to identify. Okay, at this point, the mitotic spindle is maximally developed. It's at its fully developed stage. And the reason this one's easy to remember is because here we will see that the microtubules of the mitotic spindle have pushed all the chromosomes to the middle of the cell. We can call this a number of things. The middle of the cell you can call the equator. And we actually, during mitosis can call this area in the middle of the cell. We can also call this the metaphase plate. All right, so in metaphase, all of your chromosomes are in the middle of the cell. M for middle, M for metaphase. All right, and so this is what you're looking at. Okay, that's the main idea with metaphase. Pretty easy to identify. Here's just another look at prophase and metaphase. All right, remember that early prophase you will still see a nucleus. Late prophase, the nucleus, you will no longer see that. Metaphase, we've got our chromosomes all lined up perfectly in the middle. Next we have anaphase. Okay, so anaphase also has an early and a late stage. So what's going on in anaphase? Well, here, centromeres of the chromosomes will split. Okay, so what's holding the chromatids together will break down. And then each chromosome, okay, is going to be pulled to the respective pole. So basically, you're going to have a whole set of genetic material going on one side of the cell and the exact same version of that going to another part of the cell at your poles. You could say a North Pole and a South Pole if you want to say that. And we're still having, now remember in mitosis we had all of our chromosomes in the middle of the cell. Well, during anaphase, it's the opposite. The whole idea with anaphase is that now we're pulling our chromosomes to opposite poles. We're now moving those away from the middle of the cell to opposing poles. Okay, so that's really the main thing that's happening in anaphase, is getting two sets of identical genetic information to opposite sides of the pole. You do have a late phase and an early phase for anaphase. And so here's a look at anaphase. All right, remember that the last phase was metaphase and everything was in the middle, but look, okay, we have now our daughter, it says here, daughter chromosomes, okay, but I mean, you know, really these are chromatids, okay, because you're getting one half of the chromosome brought to each respective pole, okay, that way you have identical genetic information on each side. So really that's what we're seeing in anaphase. Okay, early anaphase, you're going to be seeing this progression of these chromatids moving away from the middle, closer to the poles. Okay, now I think I forgot to mention, so yeah, I did. Well, what's really the difference between early anaphase and late anaphase? Well, really the big difference is that in late anaphase, you start to see this really important structure called a cleavage furrow. You start to see that. And that's telling you that the cell is really starting to prepare to physically divide into two. You're going to next have telophase. Okay, at this point, okay, your genetic information is at each pole. Okay. And what you're going to start to see in telophase is the redevelopment of nuclei. Since we now have two sets of identical genetic information, we need to encircle each set with a nucleus. Okay, so we will now build new nuclei. So we're going to build two nuclei. What does that? The rough ER. The rough ER is going to actually be responsible for creating the constituents of the nuclear envelope. So once we have our nuclear envelope formed, then yeah, we pretty much have a nucleus. Okay, and so this is going to envelop each set of genetic material. We don't need the mitotic spindle anymore, so that will be gone. That will start to disintegrate. You will also see the development of nucleoli within each nucleus. All right. So here is another picture of this. So in anaphase, remember that in anaphase, we're pulling these chromatids. Now in this picture, it does say chromatids. Good. All right. So remember that in anaphase, we're now pulling the sister chromatids away from the middle, and we're now bringing them to opposite poles. And then in late anaphase, this one really looks like late anaphase, because now what we're seeing is this structure here in the middle called a cleavage furrow. Okay, remember that you first start to see this in late anaphase. All right, we're going to start then to see telophase, and this is where we're going to have our nuclei totally seen now. We're going to see these nuclei, new nuclei surrounding it. Now this picture is actually showing you sort of a combination of stages. In this picture, this is showing you telophase, but it's also showing you cytokinesis. What is cytokinesis? Cytokinesis is when this cleavage furrow goes all the way to the middle on both sides, it basically pinches off the cell into two cells. That's called cytokinesis, the actual physical division of the cell. Right, so yeah, cytokinesis is actually when you have division of the cytoplasm and you have two cells. All right, so just some summary information here about the cell cycle. Okay, a couple of important points on this slide. So yeah, cell division is highly regulated, very highly regulated. We should only be doing this when we're supposed to be doing this and no other time. All right, we need to know when to divide and when to stop dividing. When do cells divide? And I've really already mentioned this, okay, for the most part. We're going to be doing this during development. And how do we know to do this? Well, the brain and certain glands in the body will release growth factors and or hormones, okay, to signal this process to start. Okay, so that's what's going on in development. When you're growing, you have a high expression of certain chemical signals called growth factors that they're released at very high rates during development so that we can continue to have our cells divide so that we can grow tissue. Now, when we become adults, release of growth factors decreases. All right, some other things that need to be in play here in order for a cell to divide. Well, we have to make sure there's enough cytoplasm for cytokinesis to actually occur. We have to make sure DNA has been replicated during interphase, during the S phase, synthesis phase of interphase. We've got to make sure these cells have enough nutrients because nutrients are going to go into what's needed to make the enzymes that we need to make all this happen. All right, and as I already mentioned, you know, your cells are also going to divide as a response to regular cell turnover, as we're going to see in the skin, or in response to injury for those tissues that do regenerate. When do cells stop dividing? Well, basically, cells like to be in contact with each other. Well, not all cell types, but some of them, such as the cells in your skin. They like to be in contact with each other. So once cells are in contact with each other, cell division will cease. When we do not have enough nutrition or when our growth factors start to decline, then we will stop dividing. And again, this whole point I was trying to make about cells being in contact with each other, they will stop once they're close to each other. This is actually called contact inhibition. You should probably know that term. You might have a question on it. So contact inhibition, okay, is when we're having cell division cease because cells have made contact with each other. All right, a little bit about checkpoints. I mentioned something about this before. So one way that we're able to regulate cell division is by way of what are called checkpoints. Now we will have several areas or points during DNA replication where DNA polymerase, for example, will check. It will check itself. It will check to, you know, make sure that the bases were paired together correctly. In other cases, checkpoints are controlled by these protein complexes called cyclin CDKs. There are many, many cyclin CDKs. I'm not going to ask that you know the individual ones or anything else about them. I'm just asking that you know the name of these complexes. Okay, so checkpoints during cell division are largely controlled by these protein complexes called cyclin CDKs. So you will see one of these complexes at G1. Remember, G1, an interphase. All right, these will come in and check to see how things are going. Okay, If DNA is replicating properly, then the cell will be permitted to continue to the next stage, which is the S phase. All right. G1 is really all about growth, though, remember. So cyclin CDK is going to come in and look, do we have all the proper machinery to do all of this? Do we have DNA polymerase fully made? That kind of thing. All right. If the cell is not ready to go to S phase, then the cell will go into another phase called a non-cycling phase, meaning that this cell will not go into mitosis. Okay, it will be put aside, and this is considered the G0 phase or the GO phase, G0. Okay, a cell that is in the G0 phase, that means it is not going to go through mitosis because it has not passed this checkpoint. It does not have what it takes to go on to the S phase. Maybe, for example, that cell did not make enough DNA polymerase. Maybe that's a reason. All right, there could be a lot of reasons. There's another checkpoint at G2 and also mitosis, okay, late in the G2 phase and early mitosis. There will be another checkpoint to figure out, is this cell able to really go through with full-blown mitosis, okay? So we do have a couple of checkpoints here, and there's actually many more checkpoints, okay? And there's so much more going on in here than this, okay? But I'm just asking that you know these main checkpoints here. All right, we're going to switch gears here a little bit and talk about some basics of heredity. All right, so heredity really is just referring to how we transmit genetic material from parent to offspring. Something called a karyotype. This is a chart where you can see all 46 chromosomes. These, on this karyotype, these chromosomes are arranged by size. Remember, okay, 46 chromosomes, that means there are going to be 23 pairs. Okay, 2 members of each pair are called homologous chromosomes. Okay, this just means we have a full chromosome. Okay, 1 chromosome of each pair is coming from each parent. Okay, so you get 1 chromosome from mom and a second one from dad. And that's how you're going to get your 46 chromosomes. 22 pairs are called autosomes. Okay, autosomes look alike and they're going to carry the same genes. Now there's another pair, remember we've got 23 pairs, there's another pair, 1 pair that are going to be the sex chromosomes. These are called X and Y. Okay, females are composed of 2X. Okay, you have a homologous. Homologous just means same. Okay, same. So you have 2X. For males, there is 1X and 1Y. Here is a picture of a karyotype. You can see that they are arranged by size. Basically, you have the largest up here, the smallest down here as chromosome 22, and then here is the XX for female and XY for male. A couple of other just definitions. Locus. Locus is just what it sounds like. It is referring to a location on a chromosome. So that locus is basically referring to the location of a certain gene, okay? And so that gene is going to code for some sort of a trait, okay? So the locus is just referring to where on that chromosome that particular gene is located. What are alleles? Alleles are different forms of a gene that are located at the same locus on 2 chromosomes. So for example, mom and dad have an allele for height, all right, how tall a person is, right? So mom has an allele for height that has a specific location on her chromosomes. The dad also has a gene for height, or an allele, okay, that's located at the same place on his corresponding chromosome. All right, so these are just called alleles. Now, let's say, for example, mom has an allele that codes for being short, but dad has an allele that codes for the trait of being tall. All right, so both genes have to do with height, yet they are different. Okay, so that's called an allele. All right, we have two genes here. One is for being tall, one is for being short. That's why they're different, okay? But they both code for height. Therefore, they're both going to be at the same location on mom's chromosome and dad's chromosomes. Now, with alleles, you're going to have a dominant or a recessive allele. The way we write these out, dominant alleles are represented by a capital letter. Recessive alleles are represented by a lowercase letter. So the dominant allele, okay, it's called dominant because this is the allele that will be expressed in the individual. And this ends up masking any effect of a recessive allele. This is what codes for a visible or observable trait in the person. What is a recessive allele? This one will not be expressed, okay? This is a form, okay, of a gene or a trait that is not going to be expressed by that individual. It is recessive. The only time an offspring will express or show a recessive trait is if both parents possess the recessive allele. Okay, so recessive alleles are typically more rare. Okay, not always though. There are some types of alleles, which I'm going to mention an example of here in a minute. There are some types of alleles that are recessive, but like they're classified as recessive, but they're more common in people for a certain type of gene. And again, I'm going to give an example of that here in a minute. Now, another point to know here is that recessive alleles often code for non-functional variants of a protein. Okay, so you might express the recessive allele, but it will not serve as a functional part of you, basically. Let's talk about the terms genotype and phenotype. Genotype is referring to the actual genetic code for a trait. All right, so this is the actual DNA code that ends up resulting in some sort of trait. Okay, so genotype, genetic code, genotype. That's how I remember it. Right, and so a genotype, okay, as the actual genetic code is going to be either homozygous or heterozygous. If an individual is homozygous, okay, they have two identical alleles for a specific trait on their chromosomes. Okay, so on each sister chromatid, they have identical alleles. Okay, let's say mom, let's talk about eye color. Let's say mom has the allele for brown eyes on one chromatid, and she has the allele for brown eyes on her second chromatid. So she has two identical alleles, both for brown eyes. That means she is homozygous, all right, for that trait or that allele. If someone is heterozygous, that means they have different alleles for that gene. Maybe dad has on one of his chromatids the allele for brown eyes, but on his other chromatid, he's got the allele for blue eyes. All right, then that means he is heterozygous. All right, that means he has different alleles for the gene of eye color. He has a brown allele and a blue allele. Okay, so homozygous, you've got two identical alleles. Heterozygous, different for the same trait. Okay, so that's how you can characterize a genotype. You can refer to the person as homozygous or heterozygous. And then actually you're going to add the words dominant and recessive to this. You can say someone is homozygous dominant, or you can say someone's homozygous recessive. Similarly, you can say someone is heterozygous dominant or heterozygous recessive. Phenotype is the other word that sort of corresponds with genotype. Phenotype is referring to the physical traits that you can actually observe or see, okay, that comes from the genotype. I remember phenotype because it starts in pH, and it is really referring to the physical trait. Okay, so pH for physical, pH for phenotype. All right, so an allele is going to be expressed. Okay, basically you can say if you observe the phenotype, then yeah, that allele was expressed. Now keep in mind that parents can be healthy, heterozygous carriers of hereditary diseases. All right, so you might have a parent that has, let's say it has a heterozygous situation where maybe diabetes, okay? Diabetes does have a hereditary component. Maybe mom on one of her chromatids has an allele that serves as a predisposition for the development of diabetes. But on the other chromatid, she doesn't. And then combined with the father, let's say the father does not have any alleles that may set him up for diabetes predisposition. Then in that case, mom was a carrier of that predisposition, yet the offspring did not receive it. Okay, the offspring did not inherit that predisposition for diabetes, but the mom is still considered a carrier. All right, if the mom got together with her second husband or whatever, and that guy was homozygous for that same predisposition, then maybe the offspring will end up with that predisposition for diabetes. Okay, so just keep in mind, heterozygous, okay, you can be heterozygous and be a carrier of a disease and not actually express the disease, but perhaps your offspring will. Now, one way to sort of look at or predict the outcome, one way to predict what kind of a traits an offspring is going to get, you can set up what's called a Punnett square, where basically you're showing the different combinations of genotypes that are possible from when you combine mom and dad. And then you can make predictions on what they're going to have. All right. Oh, so here's a little example, something kind of interesting. Here on the left, all right, this is a picture of John Travolta, and he has a cleft chin. So it looks like a little dimple in his chin. You know people that have that. And then here's Arnold on the right. He does not have a cleft chin. Okay, now look here. We have our alleles written out here. Okay, so cleft chin, what is that? Well, let's look here at this at the bottom. This is a Punnett square. So what we're basically trying to predict is this person going to get the cleft chin or not? Up here in this first box, okay, we're showing capital C and capital C. That means we have a homozygous situation. Capital C and capital C. That means we're homozygous dominant. Homozygous meaning both are the same. You have two capital C's. They're the same. Homozygous. All right, now look down here. We have a heterozygous situation here, or a heterozygous combination. Now remember, these are all just the 4 possibilities that can result. You can have here at the bottom a heterozygous situation where you have one dominant allele for cleft chin, and then you see a small case C next to it. That means that on one chromatid we have a dominant trait for this, and then on the second chromatid we have a recessive trait for this. Let's look at another possibility up top. Heterozygous, okay? Same thing. Now notice that anytime you have a capital letter, so anytime you have a dominant allele, then that is what will be expressed. So what this is showing you is that the cleft chin is actually a dominant trait. It's actually a dominant trait. Because if you look here in the capital C situation, look, he's expressing it. If you look here at the capital C, lowercase c, we're heterozygous, but we're heterozygous dominant, meaning that it will be expressed because there is a capital C here. We do have a dominant allele. Okay, the dominant allele, remember, is always the one that's expressed. All right. Now look down here. There's only one possibility here of not having a cleft chin, and that is in a homozygous recessive situation. So think about that. Not having a cleft chin is actually a recessive trait. Very interesting. And this is kind of like the example I was hinting at earlier, is that this is an example, okay, of a trait that is the not having a cleft chin is an example of a homozygous recessive trait, meaning that you're only going to not have an uncleft chin if both parents are homozygous recessive. Okay? Very interesting. And there are some genes out there like that where it is the recessive one that will be inherited. Okay? But in most cases, you know, the more common trait is going to be homozygous. All right, but that was kind of like a Punnett square. I'm not going to ask you guys to do Punnett squares. I feel like, again, you know, I don't want to get too involved in this material because I really want to get more into anatomy and physiology and get into that fun stuff. This is all, you know, chapter 1, chapter 2, and chapter 3 were very much of like an introduction to understanding some things that we get into later with physiology. All right, we're going to wrap up here shortly. What is sex linkage? Okay, so sex-linked traits. This means that a certain gene or a certain trait is carried on the X or Y chromosome. And so because of this, then what this means is that one specific gender is going to receive the trait from mom or dad. Example would be like with color blindness. Color blindness is considered a sex-linked trait. Okay, the reason for this, well, the recessive allele for color blindness is on the X chromosome, but we don't have an allele for this on the Y chromosome. Okay, so what this means is color blindness is going to be passed on from the mother, okay, because mother has X. So this means that if someone is colorblind, born with that, that means mom had the allele for color blindness on her X chromosome. But keep in mind that Y does not contain a gene for this. So this means that the resulting offspring, if that resulting offspring is a male, right, because male is made of X and Y, female is made of XX. So if a mother gives birth to a son, and that son it does end up expressing this recessive allele on their X, they will express color blindness. Okay, so color blindness is often carried by the mother but expressed by the son. Okay, so if the mom has a daughter, the daughter will not express it because she doesn't get Y chromosome. Okay, so here is an example where a parent is a carrier, but they do not express the disease. The son or daughter will express, okay, the disease. If it's color blindness in particular, it's usually going to be the son. Now, flip the coin. Sex-linked traits can also occur where the father carries something on his X chromosome, but he may not express it because it was recessive in him. But he may have a daughter and pass it on to the daughter, okay, even if the mom does not have that trait. So sex-linked traits can occur on the other side of the coin as well, where the father may transmit just to the daughter, okay, or just to the son. Okay, so it can go any of these ways, but I will tell you what's most common. Sex-linked traits are more common where the mother is carrying the trait and the son is going to express it. So some other sex-linked disorders, there are many of them actually, but here are just some other examples, and I do think you should know these examples. We're not going into detail with the pathology of these diseases, except for Duchenne muscular dystrophy. We will talk about that at some point. But just so you know, these are some examples. Hemophilia, which is a blood clotting disorder. And again, this is going to be expressed much more often in males. All right, the mother carries the mutation for this. And so it ends up, she carries it, doesn't express it, but then gives it to her son who will express it. Duchenne muscular dystrophy, same thing. Duchenne muscular dystrophy is a muscle disorder, which is really interesting pathologically, and we will talk about that when we get into muscle. Another one called Fragile X syndrome. Fragile X syndrome, this is really the cause to mental retardation. I actually studied this disease when I was a neuroscientist. And yes, there is a link here between mom carrying it and passing it on, okay, to the X chromosome of her son. Okay, that's why we call it Fragile X. And here's a sort of a visual of that, right? So let's say we have our female. This is really indicate an example of the color blindness, right? So here is mom, and of course, so she's got 2 X chromosomes. All right, and then up here is the locus or location for the allele for eye color, or I'm sorry, not eye color. Whether or not a person is color blind. All right, so this is what normal vision looks like here, okay? But look at, here's a male, all right, and their Y that they get is not expressing this color blindness trait, or I'm sorry, is not expressing the normal vision trait on their Y. So they may end up with color blindness if they get this from the mother. This is the last slide, and just kind of pointing out the importance of the environment in basically DNA. I mean, some people ask the question, can you change your DNA? And, that's kind of a controversial question. Some scientists will say, no, you can't change your DNA. Others will say, yes, you can change your DNA. And I absolutely am in the camp of people that believes that you can change your DNA. Absolutely. Yeah, DNA is very changeable. And a lot of that comes from our environment, okay? example here is diet. So what you eat certainly dictates a lot of how your physiology is going to work. So let's say, for example, from your diet, let's say you're eating meat, okay? Let's say you are a meat eater. Well, then if you are, you're going to get plenty of amino acids. You're going to get amino acids because what's meat made of? What is a hamburger or steak? Well, that's meat. That is protein. And if you break your protein down, you get amino acids. So you had a hamburger today, and yeah, so you got your protein. You got this amino acid called phenylalanine. All right, you have enzymes, which are proteins. You need your meat for that too. That enzyme's going to come in and cause phenylalanine to turn into tyrosine. Oh, by the way, you don't need to know the names of these amino acids, okay? This is just showing you basically like you need these amino acids, okay, to form certain proteins. Tyrosine will end up being converted into something called melanin, which we're going to talk a lot about. Melanin is actually a pigment that gives you brown eyes, all right? It also provides a darker tone to the skin. We're going to talk a lot about melanin, all right? And so, yeah. All right, so you see the pattern here, okay? What you eat absolutely determines what proteins you will express. All right, so yeah, this absolutely affects your DNA. Absolutely. Okay, so I think that's the end. That went on a little longer than I was expecting to, but yeah, we're finished with chapter 4. So that's everything you need for that chapter, and we're going to get ready to move on next to tissues. We're going to start talking about tissues. Okay, so that's it. Have a great day.